All right, let's recap. Let's recap where we left off. We were looking at a lot of anatomy of the cell structure and then adding to these images the function of those things. And so looking at my guided notes, I was relying a lot on the structure in your guided notes because I really think it's important to have an idea about these structural differences of your skeletal muscles. So let's just go back and review. The first thing is we said a muscle that you're naming in anatomy is really called a what? An entire organ. Very good. And we talked about how our muscle organs are arranged in bundles. Technically, you'd call it a fascicle. And within the fascicles are individual muscle cells. And we said cells could otherwise be called what? Muscle cells could be also called fibers. Okay. Some other specializations about the skeletal muscle fiber is we said we could say skeletal muscle plasma membrane, or we could say, not yet, the entire sarcolemma. Okay, but the prefix sarco, right? We're going to see on things like the sarcoplasmic reticulum or the SR. What was unique about the sarcolemma? What did we say? What's interesting or unique here? The sarcolemma. Did I say the sarcolemma? What is it? It's a plasma membrane. Yes. It has voltage-gated ion channels. It retains the ability to conduct action potentials. So our skeletal muscle cell membranes are conductive, just like which type of cells? What other cells can conduct action potentials? Nerve cells. Okay. So the surface of the muscle cell is kind of analogous to the axon of a nerve cell in that it can conduct action potentials. We also noted that our skeletal muscles are multinucleated. They have multiple nuclei. That's a developmental thing that happens when we grow. Multiple cells fuse together. And this is interesting, hopefully, in that if a skeletal muscle cell is damaged, it doesn't have the ability to do what? If it has a bunch of nuclei in it, what can it probably not do correctly? What's in the nucleus? DNA, and so therefore it would not have the ability to replicate. So if you lose skeletal muscle cells due to injury, those cells are gone after you reach a certain level of development. So our skeletal muscles are multinucleated. The sarcolemma is conductive. The sarcolemma extends into the plasma, into the cell cytoplasm. What are those called? T-tubules, transverse tubules. They're also conductive too, right? They also have the ability to conduct action potentials. And they're adjacent to a specialized organelle, the sarcoplasmic reticulum, and we said we could call it the SR, which is just a specific term for which organelle? The smooth ER. It's getting its own name here because it's huge, it's extensive, and it does a really important job for the muscle. What does it do? Calcium storage and release. And what did we say is characteristic about the SR that allows it to release calcium? It has voltage-gated calcium channels. We also talked about the role of mitochondria. Right? We're familiar with that. Right? Chapter 3, Unit 1. But then we also talked about the role of these things. What is this thing? Myofibril, myofiber, right? Whatever. Myofibril. What is that? What is the myofibril?
Contracting machinery.
The contracting machinery. It's made predominantly of which type of molecules? Proteins. and contractile proteins, because it contracts. And the repeating subunit is called the sarcomere, M-E-R-E, sarcomere. And that's about where we left off last time that we met. So we're going to go through and label the regions of a sarcomere. What questions do you have about basic cells? Oh, yeah, let's not flip yet. What is this area called? Did we talk about this?
Motor end plate.
Very good. The motor end plate. That's characteristic because it has the nicotinic receptors. Good job. Which allow the muscle cell to do what? Depolarize. You guys are doing good. Making me proud. Good job. All right. All right, y'all ready to transition? I'm going to refer to figure 12.3 to label the regions of a sarcomere. And then after presenting it yesterday, realizing this presents a best picture. It's kind of like a circular, it's trying to show something in a three-dimensional, something on a two-dimensional flat piece of paper. So we'll just do our best to label the different regions of the sarcomere. I'll first turn your attention to this grayscale image. I don't have it on your guided notes, but it's certainly in your textbook. This is, on the lower end, a transmission electron microscope image, a TEM. You've probably heard of that before, which gives us grayscale. And this is an exceptional TEM image of a sarcomere where we can distinctly see what we're going to label as the Z bands and the dark and the light areas. And so we're going to take this grayscale and translate it to this kind of cartoon, colorful image and look at the different regions. So a sarcomere is the repeating unit of our myofibrils. We studied that last time. So sarcomere is what we're looking at. Our sarcomeres are microscopic and they extend from what's called Z-line to Z-line. In this case, the little blue, purple colored stuff. The Z-lines get their name because they look like a zigzag. Can you see how it's zigzagging? So I have a Z-line. So between those 2 points would be a single what? Between the 2 Z-lines, there would be a single sarcomere. Okay? And then I have another one. So there's at least 2 entire sarcomeres shown in the image above. So a sarcomere, smallest functional unit of a skeletal muscle. This is where contraction is going to happen at the microscopic level. And it is, right, extends anatomically from Z-line to Z-line. In the middle of each sarcomere is the M line, the midline. You'll notice that it's kind of shown red here, running perpendicular right to the rest of the sarcomere. And so right in the middle is the M line, the midline. There's one here and another one here. Oops. On the M line, if you notice running perpendicular to it, we have this barbed looking something that looks barbed in this image, right? Bless you. That's going to be what we'll call the thick filaments. The thick filaments branch off of the what? The which one? M line. Very good. The thick filaments are branching off of the M line. They would actually be attached right to the M line. So the M line is the point of attachment for our thick filaments. The M line is a point of attachment for the thick filaments. The thin filaments are the purple extensions off of the Z line. So the thin filaments are attached to the Z line. The Z line is the point of attachment for our thin filaments. line is the point of attachment for our thick filaments. The Z line, the point of attachment for our thin filaments. The rest of the analysis that we're going to make is dependent upon how those thin and thick filaments actually interact and overlap one another. Because if we're looking, right, these are studied with microscopes, whether they be light or electron microscopes. In any area, if you're looking at a tissue in a microscope where there's more of something, less light or less electrons can go through. And where there's less of something, more light or more electrons could go through. Okay? So that affects how we see what we see. And that's how this naming came about. And so we're going to look at her including dark and light bands. Okay? That makes it dark. And that ties into what we said muscles are striated. Okay? So we're going to have dark, light. The areas that are dark, would they have more or less of these filaments in there? They'd actually have more. They'd be more densely packed, less light going through. And the areas that visually appear light would have less amount of proteins packed in there. And so the dark area is the area where you're going to see basically the thick filaments. Wherever you see there's thick filaments, it will look how? Dark. Let me come back. All right. The green is what I have isolated as the dark. The dark area you can shorthand with A band. A, the first vowel, dark. Okay. So here, just highlight around where you see the thick filaments and label that the what? You can call it the A band or the dark band. So everywhere where the thick filaments are will appear dark. Okay. Around the Z line, there may not be any thick filaments projecting into there. So that area would look how? Light. And we'll call that the I band, the first vowel I, light. You call it the I band or the light band. What color is that highlighted here? The purple thing. Okay. So I have a light band shown in purple, a dark band highlighted in green, and the next one would be a light, then a dark, then a light, then a dark. Okay. That gives us the striation, the striped appearance. The I band is just around the what? Z line where there's no thick filaments projecting in. Around the M line, there's the dark band. Around the entire thing, light, dark, light. So we just keep going as long as that muscle is long. If you really scrutinize it, though, you're like, don't I see some light in there? You see it? Right in the middle of the dark band, it looks a little bit lighter. But it's not as light as the light band. Okay, so we're still around the M line. And if you notice, right around the M line, it gets a little bit lighter in the middle. Okay. This is here in the middle. Notice how on the M line you have your thick filaments, but no what? There's thick filaments, but no thin filaments. Okay, so right in the middle of the thick band, where there's just thick filaments and no thin filaments projecting in, it looks a little bit lighter. That area that's discernible is called the H zone. I have no idea what the nomenclature is on that. But right in the middle, right here, the H zone, right in the middle of the A band, where it looks slightly less densely packed. It's called the H zone. So that's the middle of the dark band where there is no thin filaments projecting in. So we have shown one highlighted here in the blue as well. So again, the H zone is in the center of the A band, or the dark band, right along the M line where there is no what? Thin filaments projecting in there. The sarcomere extends from Z line to Z line. And around the Z lines we have which band? The I band, and then we would have another dark, etc. This is a sarcomere. We're looking at it cut long. You can see those striations. What questions do you have about this really microscopic structure of a myofibril, looking at the structure of a sarcomere? When we get today in just a little bit, we'll look at even closer in magnification and how those thin and thick filaments actually work together for contraction. So if we look at the thin filament structure, let me just look at the word first. Filament just means what? Kind of like a tube or a strand, right? A string. We're looking at proteins that are filaments. Okay, so they're long proteins. The thin filament gets its name. It's a filamentous protein. It's long, but it's very thin. In contrast to the other protein that we're going to study. It's significantly thinner in diameter. The thin filament, the name is kind of tricky in that it's not very simply assembled, as you noticed in your handout. The thin filament is actually comprised of 3 types of proteins. The primary protein which makes up the thin filament is which? Which protein is the bulk of our thin filaments? Actin, very good. And what color is actin shown in this image? The purple-blue thing, right? Okay. All these little spherical proteins, the little blue things, are actin filaments.
They call them monomers or something?
Yes, the monomers.
One
actin could polymerize with another and make a polymer. You could go back to your, like when you did gym bio, your monomer subunit of, right, so just the building block. The assembly of the thin filament is made up of monomers of actin. They're polymerized to make a thin filament. So actin molecules, right, serve as the foundation for the thin filament. Characteristic to the actin is that on the protein, it has a binding site for another protein called what? Myosin. It's like a receptor. So there's a protein, it's like a receptor. It receives myosin. Okay? And so I've highlighted, they're shown black in the image, and I'm just showing the yellow to hopefully highlight it to you, what we're talking about. These myosin binding sites are covered up by what? Something called tropomyosin. What color is tropomyosin in this image? The pink, lavendery, filamentous, stringy-looking protein. Filamentous. Tropomyosin. What is its job? It doesn't hold the actin, it
covers those binding sites until they're turned.
Very good. It blocks those myosin sites that we just mentioned on the actin as identified until something else happens. We'll clarify that in just a second. But they have a blocking role. They bind up those receptor sites. So tropomyosin is covering up the myosin binding. We all heard that. if tropomyocin's binding, covering up the myosin binding sites, actin can't bind. It can't bind to myosin.
Oh,
Is it proper to call them strands?
I would call them filaments. I said strands just kind of conjure up some image, but they're called filaments. Alright, so the bulk of the thin filament is a protein called actin, which each little, we'll say, monomer subunit has a myosin binding site. And then we throw in there, well, there's this protein on top of it, covers it up. Well, what's the point of that? Well, the point of it is for what? Why would we express something and then cover it up?
Because it's not time for it.
It's not time. What else are you going to say? So it can return. We can regulate it, right? So if we cover it, we can regulate when the proteins interact, when the actin and myosin interact. So when tropomyosin is laying over those binding sites, the thin filament and thick filament aren't going to touch. And the muscle's not going to contract. So maybe you want to identify, if it's covering the myosin binding sites, the muscle is in what state? Relaxed state. And so we've got actin with myosin binding sites, and then another protein called tropomyosin, which is blocking that receptor site. If it's blocking it and serving a regulatory role, we say, well, there's got to be something to move it out of the way. The something that can move it out of the way is troponin. Very good. The third protein of the third filament, which is troponin. You'll notice it says it's a complex. It actually has 3 little subunits to it. So troponin is the third protein of our thin filaments. It clearly is going to bind, I don't know if it's clear or not, but it binds to actin, it binds to tropomyosin, it also binds to what? Calcium. Very good. Each troponin molecule has a receptor for calcium. And I've tried to highlight that here in the little green dot. Okay. All right. So again, the 3 proteins of our thin filament include actin, with its myosin binding sites. We have tropomyosin with its kind of inhibitory role. And then we have troponin, which is going to have more of a regulatory role. When which cation is present? Calcium. And you know where it's coming from. From where?
That's all.
From the sarcoplasmic reticulum. And so what happens is, if calcium's available, it binds to troponin. We just change that molecule's shape, structure. We just changed its function. Okay. So what happens when calcium binds to troponin, it pulls back what? What's it pulling back? Tropomyosin. Doubly expose the binding sites on the actin molecules. So I'm just going to repeat to kind of let it sink in. In the thin filaments, the primary, right, the contractile protein in there is called actin. And it happens to have binding sites for myosin, which are covered up under relaxed state by a molecule called tropomyosin. When getting ready for contraction, calcium from the SR can bind to troponin, which physically changes the status of this thin filament to expose the binding sites. So now, thick filament right next to it can bind to the thin filament to get ready for contraction. What questions do you have at this point about this kind of simplified anatomy of a single thin filament? How many proteins make up the thin filament? 3. They are actin, Very good. Actin, tropomyosin, and troponin. All right, let's contrast the thin filament now to our thick filaments. The thick filament is made up of a molecule called myosin. And then here, isolated is a single little molecule of myosin. And we'll look at a complex and actual thick filament. But if we look at a myosin molecule, you'll notice that it's complex and that its tail is filamentous. The tail is a filament. In the heads, what term do we use that? Proteins are either filaments or, what's the other word? The question was, our proteins are either filaments or anatomically they're a strand, or they're like an actin, they're a sphere, and you call them globular. So myosin has you would say, what kind of tail? Filamentous tail and a globular head. So it's a complex protein. It's not just a strand, it's just not a glob, it's both. All right, so there's the tail, there's the head and tail. And what we're going to focus on, the machinery is going to be acting at the head, where it certainly is going to have the ability to bind what? Actin. So actin has a myosin receptor, myosin has an actin receptor, so they're going to have the ability to touch, bind, interact with one another. But you also noted from your handout where there is also what here? ATP sites. So not only is calcium necessary for contraction, but also ATP. It takes energy to power muscles. Goes back to chapter 3 discussion. To do work, we need energy. And for skeletal muscle contraction, the energy comes from ATP. So again, the thick filament is comprised of a protein called myosin, which its globular head has the ability to bind with actin, and then we can go look at actual contraction if ATP is available. Those mechanics we'll get to today. But we need to understand the basic anatomy first. All right. Questions so far? Are we okay? Thin filament, thick filament? So that's the, yes? Yes, that's a good question. Is it a couple? Yes. A functional myosin molecule is not one but two of these filamentous tail globular structures bound together. And so if you look closely, you can see the tail intertwining. So that's two subunits, two heads. And so it's two, making up a single molecule, myosin. However, the actual thick filament is lots of those bound together. So they would make a big, large superstructure. But to understand the superstructure, we're just kind of picking the tiniest part apart. But you're right, a myosin molecule is a dimer, meaning two of the same type of proteins working together. So figure 12.5 will hopefully address the question that you had. That yes, there are two heads on a single myosin molecule, but then to make up the actual thick filament, they take a lot of these and bundle them together. So maybe a visual, I don't know if it's good or not, but think about a straw. I could talk about the anatomy, like a bending straw, right? I could take bend the head and bend it, right? And then if I want to look at how a bunch of them bundled together, right? Hold them together and they would all have their little heads branching off. So kind of like that. So when all the straws are bound together, it would be the thick filament. To understand that, we would take a single straw and pick apart its anatomy. Good question. Additional questions? All right. So we have an idea about the thin filament structure. We have an idea about the thick filament structure. And then we can put it in concert a little bit to talk about something called the sliding filament model of contraction. And the sliding filament model illustrates for us how the relationship, maybe the status of the A band and the I band change during contraction. And how the filaments slide past one another. They slide past one another. They don't jumble up or accordion. Maybe that's another way to say it. The thin and thick filaments slide past one another. They don't shorten. The whole thing doesn't shorten. I'm going to probably better demonstrate this if you do a hands-on demonstration. So take your hands. Okay. Your hands are now thin and thick filaments. Okay. Your thumbs are going to be your Z lines. Okay. What happens during between relaxation and contraction is your filaments, your thin and thick filaments, your fingers will slide past one another during contraction. So during contraction, what happens to the Z lines? They get closer together. But did I change the status of my fingers? Did I bend them or intertwine them? So contract, relax, contract, relax. That's pretty analogous to what's happening with the sarcomere to illustrate the sliding filament model. Is that the filaments slide past one another. Okay? They overlap more during contraction and overlap less during relaxation. Yes?
Is it true that the H zone is shortened?
Yes. Okay, so you can do the during contraction, and what happens to the I band? It shortens, the A band becomes more apparent, and the H zone may be even not even visible at all, depending on the status of contraction. So in this image that you have, this first scenario, the muscle is doing what? Relax. In the second scenario, it is contracting. So let's just go ahead and practice. I want you to highlight the A band, the I band, and I guess we can do the H zone as well. In each the contracted versus the relaxed state. So label on here the A band, I band, H zone. And each are contracted versus our relaxed versus contracted state. And then compare the relative distance. You don't have to get a ruler out, but hopefully you can visually annotate. Labeling the A band, the I band, and the H zone in the relaxed and the contracted state to see the relationship of those in a relaxed versus contracted muscle or sarcomere. So during relaxation, I'll highlight, what band did I just highlight? A. The A band, and deep in the middle is our H zone. And on the adjacent side would be the I band. Straight line. Got my I. And so where are we seeing the impact at? The H zone, and even in the, can you see in the I band a little bit? So it looks a little bit smaller compared. So yes, the visible distance does change, whether a sarcomere is undergoing relaxation or contraction. So again, the sliding filament model just tells us how these thin and thick filaments slide past one another. So the Z lines become closer to one another, and within the A band, the H zone becomes less apparent, and the I band also becoming less apparent. Any questions on the sliding filament model? Okay. And then figure 12.6b shows real images, real transmission electron microscope images, noticing how the A band less apparent, I keep misspeaking, the I band less apparent, and then the H zone significantly decreased. So again, that's figure 12.6b in your textbook. The next thing that we're going to study is the actual events that happen when a skeletal muscle gets excited. This will tie into your handout that you're going to submit today about the excitation, contraction, coupling. So we're going to add quite a bit of detail here. You might want to review your narrative that you wrote in your handout with what we're going to add here to this image. Also, it will align with the video that was shown in lab in the Mastering A&P study area, the A&P flicks. And the second video, the excitation, contraction, coupling events. And it'll tie in the anatomy of everything that we just studied. Okay, so we're just going to hopefully make sense of all the structures that we've introduced. Here we're going to say contraction occurs in just a minute, and then we'll step back and look at the actual events of contraction. Okay, so we'll just say it happens, and then we'll detail it. Everything's detailed. Someone has put a lot of time into studying muscles. They've got a lot of information for us. So what is this whole area that I'm at? What am I looking at?
Neuromuscular junction.
Very good. A neuromuscular junction is the entire thing. In yellow, we have what kind of neuron? Motor neuron, somatic nerve. Very good. Then our somatic nerves release acetylcholine. So acetylcholine is released from our somatic nerves, our efferent nervous system. And I know the text is small. I try to increase it as best I could to fit up here. But for detail, you can always turn to figure 12.8 or your narrative that you wrote in your guide notes, or your handout, excuse me. So we know that when the somatic nervous system wants to tell a skeletal muscle to contract, it's going to release acetylcholine. You also know that at the, where am I at? Motor end plate, the skeletal muscle has the nicotinic receptors. So somatic nerve, when getting ready to voluntarily tell a skeletal muscle to contract, the somatic nerve releases acetylcholine. Acetylcholine diffuses across that little junction, or you can call it a synapse, to bind with our nicotinic receptors at the muscle cell's motor end plate. You should be very familiar with the nicotinic receptors. We introduced them in Chapter 5. We know that they're just chemically gated sodium channels. They're more permeable to sodium than they are potassium. So in the presence of acetylcholine, what's the effect at the skeletal muscle? We got a long way before we get to contraction, right? Not even there yet. And action potential will happen, but first we gotta depolarize. We gotta grade up to threshold. So it's those nicotinic receptors that allow the muscle cell to grade up to threshold. Through the influx of which ion? Sodium ion. The next thing that I have shown here are points and maybe kind of blend in. That graded potential, I could say a graded potential at the motor end plate, an alternative name is called an end plate potential. So the nicotinic receptors, when activated, cause the muscle cell to grade up to threshold. Then we can generate an action potential. So again, I'm going to restate that. Acetylcholine binding at the nicotinic receptors generates an end plate potential. Which applies all that back stuff I just said. Influx of sodium, grading up the threshold, right? All that. Or I could have said end plate potential. Initiates an action potential where? Along the sarcolemma. We'll go, it is the membrane, but we'll go with sarcolemma. Okay, so an action potential is conducted along the sarcolemma and down the transverse tubules, the T tubules, colored, which color here? Green. An action potential is conducted. You're going to run over. And maybe if it's going to help you put this together, draw some voltage-gated channels along the sarcolemma and some voltage-gated channels along that T tubule. Just put some little, like I've drawn in the past, I like to put a little gate in there.
So at this point, are all of the voltage-gated channels for sodium?
What was your
the membrane and on the TTV, are they all sodium, or are there other ones?
It's predominantly sodium. There's actually other ones, but I'm not going to throw that out there. There are other ion channels. They don't contribute to the action potential. So we're just, may not feel like it, but we're putting our blinders on. Action potential. Voltage-gated sodium, voltage-gated potassium channels. Okay? Good question. So again, stepping back to put it together, somatic nerve releases acetylcholine, diffuses across to bind with nicotinic receptors, which lead to an end plate potential, which initiates an action potential along the sarcolemma and down our T tubule. Still can't say contraction yet, can we? No. Nope. All right. Action potential affects the permeability of action potential along the T tubules affects the permeability of the SR, okay? The voltage-gated calcium channels on the sarcoplasmic reticulum. Okay? So that action potential opens up the voltage-gated channels in our sarcoplasmic reticulum. Calcium is released to the cytosol. Very good. All right, so calcium goes out to the cytosol. And you know now what? It's available to bind to. Where's calcium bind? Troponin of our thin filament. So now that calcium's leaked from the SR, it's in the muscle cell cytosol, and it binds to troponin. We identified that causes a physical change to expose our myosin binding sites of our, where are the myosin? It's actually in the thin filament. The myosin binding sites are on the actin of the thin filament. Reciprocal. That is going to lead to contraction, which I said we'll come to in just a minute. But muscle cell contractions don't last indefinitely, right? All things must come to an end, including muscle cell contraction, which is good. And so, what should happen?
Calcium's kicking back.
Very good. Put that calcium back into the sarcoplasmic reticulum. Right back in. And once calcium's back in the sarcoplasmic reticulum, the muscle should relax. The muscle cell should be able to contract when calcium's in the cytosol, but if it's out of the cytosol, it's going to relax. The contraction, the events are summarized in what's called the cross-bridge cycle, which we're going to talk about coming up. Yes, during the cross-bridge cycle, you want to put in the parentheses contraction. In addition to reviewing figure 12.8, again, go back to the master AMP Flex video for excitation contraction coupling. We'll put it in concert for you, hopefully. All right, so y'all want to repeat it again?
Yes.
All right, let's go back through. In order to get a skeletal muscle ready for a contraction, we first have to excite it. And so we got these events called excitation, contraction, coupling. You notice it's not real simple, right? It's really fast, though. So what happens is this somatic nerve will quickly release acetylcholine. It diffuses across the cleft to bind with nicotinic receptors, which cause the muscle cell to grade up, or you could say generate an inflate potential. It grades up the threshold to generate an action potential, which is conducted not only across the surface of the sarcolemma, but also along the T-tubules, which takes it from the surface to the interior. So we're conducting intracellular action potentials. That's good, because right next to our T-tubules are things called the SR, the sarcoplasmic reticulum. It, in response to an action potential, will release calcium into the muscle cell's cytosol, which can then bind to troponin of the thin filament. Troponin's on the thin filament. Calcium is a messenger. The messenger is to do what? Expose those myosin binding sites of actin so that actin and myosin can touch. They can bind one another. Whole series of reactions happen, which I just said contraction. Okay, so we're going to come back and clarify the events of contraction. And then once the muscle cell is told to relax, what happens? Calcium's pumped back into the SR, troponin, tropomyosin, cover up, and the muscle should relax. Those events are collectively called excitation, contraction, coupling. What questions do you have about that at this point? All right, so let's look at the detailed events of contraction then. It's collectively called this cross-bridge cycle. Oh, all right, so I'm just going to refer to figure 12.7. We're looking at that thin and thick filament touching one another. It's a cycle, right? Touch, ratchet, let go. And so it looks like your image is gonna start kind of the lower, right, around 3 o'clock, right, in the image. Just to put the image in perspective, we have our thick filament in the beigey color, the thin filament in the blue complex. Okay? You should be able to see the myosin binding sites. We don't actually have them covered up. So our myosin binding sites are exposed, colored in the black. And then the calcium is shown in green, the troponin in the globular pink, tropomyosin in the filament is purple. So they're keeping the same color scheme. Are you okay with or really super microscopically, down to the molecular level. So when calcium's available, you acknowledge that troponin pulls back tropomyosin to expose the binding sites. And so what happens is, as the name implies, actin and myosin bind. So they just physically touch one another. But contraction involves movement and force transferring. So we got to play into this is a source of energy. And our source of energy comes from which molecule? ATP. You notice that we don't just have ATP here. We'll get there, but where is it bound? On its binding site on the head of the thick filament, the myosin. ATP, if you remember, can be made from ADP and inorganic phosphate. The P sub I is our nomenclature for inorganic phosphate. So at this point, just the thin and thick filaments are touching. And now we want to, if you remember, we want to slide these thin filaments over the thick filaments. So it's going to take energy. And what happens is, after ATP is available, first we're going to see inorganic phosphate removed. So inorganic phosphate is released. So what is still bound to the myosin head? ADP. And when that does that, look at the state of that globular head. Inorganic phosphate present, inorganic phosphate absent. Can you notice the status change? That cross bridge just basically flipped, if you will. Just transferred out energy and it changed its shape.
The head.
The head, yes. The tail and everything's securing it in place, and so the head is doing the work, if you will. ATP split, and then inorganic phosphate removed. But remember, it's bound to actin, so when it changes state, what's happening to the thin filament? It moves, right, to the center, right, towards the M line. Then, what is going to be released? ADP is going to be released. That is referred to as the power stroke that I was trying to illustrate. Okay, it's called the power strokes. We're moving and transferring power. It's moved in by a single actin molecule distance. And then ADP is released. Once ADP is released, notice that the cross bridges are still bound because what's still present? Calcium is still present. So it just did like this. So think of, I don't know, I always think of like tug of war. Think of the rope being like the thin filament and your arms being the myosin cross bridge. So just think if you had one hand, reach out, pull. You're still touching. And then I have my other one, my other head was addressed, right? But before I can pull again, what do I need to do? Let go. First one needs to let go. It's not going to happen in the myosin cross bridge until a new what is added? New molecule ATP will cause those to quickly dissociate. I'll come back to that word. A new molecule will bind to it and causes that myosin cross bridge to do what? Let go, unbind. And now, since there's a new molecule of ATP available, we can split it and what? Reattach. If there's still what available? Calcium. And then we just power right through. Just bring in. So it's imperative that if we want the thin and thick filaments to detach from one another, the thick filament myosin head needs to receive a molecule of ATP. So it's called the unbinding. Or you could say, right, it's As soon as it binds, it then splits and resets the system. So new binding of ATP dissociates the thin and thick filaments. It splits and kind of resets the system.
So how this keeps cycling till relaxation?
This will all keep cycling until relaxation or fatigue.
Now, is that why you shake when you're holding, say, heavy weights? And you start shaking?
Yeah, so if you fatigue your muscles, if you're holding something, and it may not necessarily have to be heavy, but you're holding it for a long period of time, like a kid or something, right? It's like, it gets right, heavy. So what happens is you're going to either run out of ATP and or calcium or a whole other list of chemical imbalances may be present that could lead to it. But the probably most likely suspect is calcium regulations off, ATP level low. But yeah, it'll keep continuing as long as ATP is being made, as long as calcium is available. It'll just keep cycling and sustaining the contraction.
If you're dead, that head stays attached.
Yes. I wanted to come back to this word, the rigor. Okay. Between the thin and thick filaments, if they're attached, they're bound one another, but there is, right, at the point before a new molecule of ATP is added, they're just stuck. Okay? They're right in the contraction, they're stuck together. To unbind it, we need a new molecule of ATP. And so you may be familiar with the rigor if you've heard of rigor mortis. Have you heard of that? What does that mean?
Where your body stiffens up after you die.
Post-mortem, the muscles, right, gain rigor. Okay? So what happens during death is, well, you're dead. You're not taking any more what? Oxygen. You're not making any more ATP. You're not maintaining homeostasis. Your SR can't pump calcium back into it. So calcium's out in your muscle cells. What? It's not in the SR, so it's in the cytosol, binding to, calcium is binding to troponin, pulling back tropomyosin so that actin and myosin can bind. But they can't unbind because there is no more ATP. So the muscles gain rigor after death. Until everything completely breaks apart. I've not had the luxury of seeing that. I don't want to call it luxury. Whatever. I've not had the opportunity. That sounds nice. Do not want to see it either. All right. So this collectively is called the cross-bridge cycle. As is implied, it is a cycle. It's continuous as long as this inputs are available.
Would the rigor be like whenever you get tetanus and it just goes untreated and all your muscles contract up?
That would be another extreme example of maintaining, right, sustaining contraction. And that is usually, well, if you think about tetanus, the bacterial infection.
Mm-hmm.
I don't forget. I can't remember the I have to look that up, but still attaching.
I just know if it goes untreated, like when you die, all your muscles contract up and your body
Diaphragm.
arches.
Yeah, that's not good. But tetanus infection is different than the term tetanus. It just means sustained contraction. Okay, so there's a difference between an infection versus the state of the muscle. But yeah, it's lethal. All right, so within that excitation, we can look at contraction. So in other words, it takes a bit of time to lead up to contraction. And therefore, if you look at a single muscle cell within an entire muscle organ, if it's excited, it won't instantly contract. Okay? And so what happens is you could label this as a muscle twitch or highlight the word twitch. As soon as the stimulus is received, does the muscle cell, as soon as acetylcholine is received, will a muscle cell immediately contract? No, we can't say the muscle cell contracts. No, we gotta grade up the threshold, we gotta generate an action potential, we gotta conduct it down the T-tubules, we gotta let calcium out, calcium's gotta bind, we gotta ATP, right? There's a ton of stuff going on. And so therefore, when a skeletal muscle receives acetylcholine, there's a bit of what's called a latent period, where there is no discernible force generated by the muscle. So if we look at this plot over time, looking at the tension of the muscle cell contraction, the force, looks like it's measuring in grams, even though the stimulus acetylcholine is present, okay, for a period of time, there is no contraction. What are we going to label this as? Latent period. Okay. That's all that excitation, contraction, coupling going on. It's taken us about 2 weeks to talk about between lab now and today. It's called the latent period. That's where that coupling is happening. Then we can generate tension. Then the muscle can actually contract. And so as the muscle contracts, you'll notice the force of contraction kind of increases, I want to use the word smoothly, gradually, fluidly. It's a nice little incline. And then the muscle will do what? Relax. You notice it just doesn't just go from all to none, right? The muscle will also kind of more progressively grade down through relaxation. It takes a while to get that calcium back into the SR and kind of reset everything. So contraction is certainly faster than the relaxation, but we also have to take into consideration that latent period. Again, this is a single muscle cell. Okay, are our muscle organs made of single cells? No. All right, so it's obviously going to be more complex if you look at an entire muscle organ. Okay. Any questions about the phases of a twitch? And so the question is, well, how can we talk about this? You could look at increasing stimulus, a single twitch. We went through the latent phase, contraction, relaxation. But in order to really have an entire organ working functionally, we've got to stack muscle contractions or muscle twitches together. And so the first scenario, the red arrows are indicating the amount and frequency of the stimulus. Okay, so one arrow, one single stimulus. The more frequent arrows, the more frequent the stimulus. And obviously there's a direct relationship. The greater and the more frequent the stimulus, what happens to the force? It's stronger and it may even be maintained. Okay. If we look at muscle contraction, where a muscle gets stimulated, it begins to do what? Relax. We can receive a new stimulus and again do what? Contract, and then maybe relax and receive a new stimulus and then contract. And our force of response is getting greater. This is called summation. I want you to kind of hone in on that word. Skeletal muscle cells can sum contractions. They can do what with them? Sum means add them together. This is unique. If you remember our nerve cells, we said we don't want our nerve action potentials to sum together. We want the action potential to go one way. So we have that little refractory period. Do we have a refractory period in our skeletal muscle? No. We can sum these excitation events. We can sum these contractions to increase the force output by the skeletal muscle. And the same thing. We can just see an increased response. We still have some summation, but we see that term tetanus. There's a difference between Clostridium tetani giving you the disease, okay, versus the term tetany for sustained contraction. If the stimulus on the skeletal muscle becomes frequent enough, where we kind of max out on the force generated, yeah, but we can still see a bit of relaxation going on, that's called tetanus, here it's described as incomplete. Or if the stimulus is so great, we're going to max out or plateau the amount of force. Notice it's the greatest height. And there is no discernible relaxation. The muscle's just completely in what you would call it, maybe in the disease case. Right here it says fused tetany. So the takeaway is that skeletal muscle contractions or twitches can be summed together, added together, and we may even max out the force generated in a term called tetanus. We'll conclude our discussion here. We'll pick up the length relationship and the rest of our skeletal muscle discussion the next time that we meet. And again, your Chapter 12 review questions will be forthcoming. And then, so review Chapter 12 over the weekend, and then go ahead and start thinking ahead for Chapter 13, cardiovascular system.